So just perfect. I'm coming back to academia after what Pierre has just said. It's exactly where we stand for 30 years later. We were working before in Aventis Pharma, before Sanofi. You mentioned another company, but there was Aventis Pharma where we worked together there to screen already a lot of lipids to provide system delivery for DNA. So all this work that had been done before is actually important and still alive and pave the route for now what we can see for mRNA. And of course, you don't see any lipids here, even though I'm supposed to talk about lipids, because they're absent, actually, of the sRNA and mRNA prize awards, let's say, that Chantal had mentioned. So, in fact, this molecule would not go inside the cells without the lipids, and cannot express themselves, let's say, without lipidic systems. So now, of course, there was a strong limitation, which was the nuclear passage when we were working on gene delivery. This nuclear membrane is a main issue, but of course, lipid nanoparticles now are far more efficient as they can deliver directly in the cytoplasm. All the work that has been done in these past years were about all those barriers to be crossed, and also the delivery of the cytoplasm. There's a lot of chemistry that has been done to design lipids which would be able to deliver sRNA or mRNA, and of course there's still work to be done which can improve still the lipids which does exist right now. In the lab, we are interested in of course this topic, and we've started some years ago the systemic delivery or study of lipid nanoparticle by implementing DOE, which is used usually for pharmaceutical technology, like have a plan of experiments that we have associated with machine learning to have the whole system of producing lipid nanoparticle and trying to be able then to select new nanoparticle, and we can see that we have a very good correlation between the prediction and the experimenter here. So our idea and our interest is really to go forward on this, and particular to design new formulation for immune cell targeting. There's not only Pierre talked about the 4 different lipids that we can find in all LNPs, but there are all the possibilities for formulation. So it's a very open field where you can mix very different systems. And for instance, here for sRNA, we had done a system with an ionic polymer to be able to stabilize the sRNA, which is released far more efficiently than the mRNA, for instance, because it's a small molecule. And we have shown that according to various administration pathways, with this type of formulation, we could really have a proof of concept, for instance, for gene, which were of interest in the case of different collaboration. So this is the work of my colleague, Virginie Escriu. We have also developed a small plasmid, which is still in the non-viral pathway, to be able to cross more easily the nuclear membrane. And this plasmid is the P4. It's free of antibiotic-resistant gene, small size, so more easy to handle. And actually, it has been tested in 15 patients right now. It's produced in GMP to restore the neuron here for patients which are implanted. So you can see here that before the gene therapy and after the treatment, there's a far better hearing performance for all these patients. So what we're interested in is performing new formulation and also using our plasmid P4, which is very easy to handle. Just other formulation, just to give you some ideas of what we can do for formulation, and it's really open. It's here with Chantal, we have done microbubbles, cationic microbubbles to deliver the P4. This is also another type of formulation, and it's really depending on the administration pathways and the pathology you want to target. So there's a lot of possibilities with these formulations. And here in this case, we wanted to have an accumulation in expression, sustained expression in the hepatocyte, and this is what we showed as we got to micromarker, which are commercialized microbubbles. In this other study here, we mixed, for instance, this P4 plasmid encoding IL-12 with the lipids which actually activate TLF4 pathways to have a better immune response together with ultrasound. So if you mix them both, then you can see that we have a better survival only if we have the complete system. And in our case, what we're looking at, we're treating the first tumor and we're looking at the distal tumor to see whether we have an immune response. So just to finish in time, this is what we actually pursuing with not only the first part, but now we implemented the cell culture, and we have a platform on in vivo imaging, on optical in vivo imaging to follow by distribution and expression of gene. So we can also have the complete evaluation, at least of the biodistribution of the vectors and the first expression according to the virus administration path. So this is what I wanted to say, and thank you for your attention.
Thank you very much, Nathalie.